Hello and welcome to this webinar titled Characterization of Host Cell Proteins Using Mass Spectrometry Enables Effective Purification Optimization, hosted by Biopharma Asia magazine and presented by Dr. Li Zang, Dr. Chongfeng Zhu and Dr. Stephen Tate. My name is Shri Patel and I will be the moderator. Now before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we will go through these at the end of the presentation. Now please allow me to introduce you to our presenter. Dr. Li Zang is currently an Associate Director managing the Protein Analytical Development Department at Biogen. Her PhD focused on development and application of highly sensitive liquid chromatography mass spectrometry technology for breast cancer biomarker discovery. After joining Biogen in 2005, Li developed her expertise in biopharmaceutical development, especially in detailed protein structure characterization using separation and mass spectrometry. She has participated in the development of a large category of biopharmaceutical programs at Biogen over the last 12 years, including monoclonal antibodies, receptor Fc fusion proteins, blood coagulation factors, bispecific antibodies, antibody drug conjugates, biosimilar programs, and small endogenous proteins. Dr. Chongfeng Zhu has a PhD in analytical chemistry from Fudan University. Later, he joined NYU Medical Center as postdoctoral researcher working on mass spectromic analysis of proteins. He has been in Biogen since 2009. His expertise includes development of analytical methods and characterization of protein therapeutics using separation methods and mass spectrometry. He has published over 30 peer-reviewed papers in scientific journals. Dr. Stephen Tate received his PhD from the University of Warwick and has worked at multiple biotech and pharmaceutical companies in assay development. Following a move to Applied Biosystems to work as a support scientist, he has spent the last 11 years working in the research for science. Projects have included assisting with the design of triple-top system platform, as well as being a pioneer of data-independent SWOT acquisition. Currently, Dr. Tate is focusing on developing software to enhance the customer experience and simplify data processing, enabling biologists to access mass spectrometry. I would now like to invite our presenters for the first half of this presentation, Dr. Li Zang and Dr. Chongfeng Zhu, to join our webinar. Welcome, Dr. Zang and Dr. Zhu.
Thank you. I'd like to thank Biopharma Asia magazine for hosting this event, and I'd like to thank all the audience for joining us today. Dr. Chongfeng Zhu and myself, we will talk about the application of LC-MS technology for advanced HCP characterization and its application on cell line and purification development. So a typical biopharmaceutical process is composed of 4 different steps described on this slide. So first, an expression cell line is chosen for the production of the biologics. The second step is the cell culture step, which is commonly known as the upstream process, followed by a downstream process to purify the product, and at the end formulating into an API, also called drug substance step. So each one of the steps has a contribution to the control of the process-related impurities, such as whole cell proteins, such as the HCP species from the chosen cell line, certainly determine the population of HCPs potentially present in the final product. The cell culture performance, such as viability, at the end of the cell culture could determine the initial concentration of the whole cell protein impurities. And the purification step, the efficiency of removal of the HCPs, determine the final HCP content in the drug substance. So commonly used mammalian cell lines include CHO cells, HEK cells, NS0 cells, and all of those cells contains more than 20,000 genes in them, and in the meantime can express up to 10,000 proteins. A commonly used proteomics methods have reported identification of 500 to 2,000 HCPs in the CHO HCCF. So HCP is a known process-related impurities and needs to be controlled to a certain level that's that's to ensure the product quality and the safety. So the reason that we need to control the HCPs is there are a few reasons for that. So HCP that's present in the final drug substance, it could potentially be immunogenic and cause some immunogenicity to the patients. And in the meantime, some HCPs could possess certain enzymatic activities that could cause degradation of the product, or the excipients such as polysorbate that's co-formulated with the drug substance that can affect the stability of the product. In the meantime, some HCPs were reported to interact with the product and therefore affect the efficacy of the product through affecting its binding to the target. So traditionally, a multi-analyte ELISA HCP assay is the workhorse for HCP monitoring for biopharmaceutical development. The assay relies on use of antibody reagent that's generally targeting all the population of the HCPs that are potentially present. So that could then report a total number of level of HCPs as a result. So that assay has good throughput and is rather user-friendly, so it has been commonly used for a while. Mass spec assay that's applied to HCP characterization can offer some complementary information on top of that ELISA HCP assay. So mass spec method is can detect all the protein, the HCP species inside present in the samples without the dependence on the reagent for detection of the HCPs. And the mass spec characterization can also provide the identity and potentially quantify the individual HCPs and provide the clearance curve of the individual HCPs during the downstream purification. The information with the identity of the HCPs, we can also gain more information about the physical chemical properties of the HCP, which could be a useful information for development of downstream purification. The identity can also provide more information regarding its potential impact on the safety and of the, such as immunogenicity. Those could be leveraged in the risk assessment of the HCPs that are present in the final drug substance of drug product. So a commonly acceptable HCP upper limit in the biologic drug substance or drug product is about 100 ppm. And in order to identify the HCPs with higher sensitivity, a lot of enrichment methods have been used. Some of them rely on affinity reagents, such as anti-HCP reagent, a proteome miner, protein A, protein G drug product depletion type of approach to enrich the HCP for identification using LC-MS. And the chemistry method using multidimensional LC chromatography, liquid chromatography suppression could also be used to enhance the dynamic range of the coverage of HCP identification. So another way of characterizing the HCP or monitoring individual HCPs is through using different modes of chemistry of mass spec methods such as SIM, PRM, and MIM, and those methods are reported to have 1 to 2 ppm LOQ for quantitation of individual HCPs. So for high fidelity HCP identification, sometimes the cell line that's chosen for expressing the protein could propose certain challenges, such as HEK cells, which is known as the human cell line. And some of the human intervention, during the sample preparation could induce human contaminants into the LC-MS workflow. That needs to be carefully studied before applying the LC-MS to the HCP characterization for HEK cell line. So starting from next slide, Dr. Chongfeng Xu will talk more about technical details and use 3 case studies to illustrate the application of HCP LC-MS to downstream and upstream process development. Please.
Okay. First, I would like to thank Dr. Li Zang for the introduction. Now I will talk about the application of 1D LC-MS to facilitate the downstream process development by characterization of the host cell proteins using 1D LC-MS. 1D LC-MS is generally considered to be a high throughput method. It can detect and quantitate host cell protein at level down to 10 to 20 ppm, so that fits very well for the downstream characterization. Showing the figure below is a typical proteomic workflow for host cell protein analysis. It starts from the digestion of the protein complex of the mAb or the protein drug, as well as the host cell proteins using enzyme, usually trypsin. The resulted peptide was separated using a reverse phase color, and the eluted peptide were ionized through electrospray ionization and detected in a high-resolution active mass spectrometer. The mass spectrometer usually operates in a data-dependent acquisition mode and is cycling between two types of scans. The first scan is so-called MS1 scan. In MS1 scan, the peptide mass and intensity information were registered, and then it goes to the second scan, so-called MS2 scan. In MS2 scan, the most response peptide detected in the MS1 scan was sequenced, and the peptide sequence information from the MS2 scan was searched against a protein sequence database to identify the host cell protein. Meanwhile, the peptide intensity information from the MS1 scan was used to perform a value quantitation of the host cell protein using the so-called high-3 semi-quantitation method. So in Biogen, analytical development has a very good working relationship with the cell culture and purification folks. We together to solve the challenge associated with host cell protein. Below, I will show you 3 case studies which are the results of this type of collaboration. The first case study is about the irreproducible host cell protein level in the final purified drug substance samples. And the second case study is about the understanding and optimization of the Pro A chromatography, which is the most powerful step for the purification of mAbs and FC fusion proteins. The third case study is about the target host cell protein removal through cell line engineering with the use of siRNA. Case 1 is about a small basic protein. It's a non-mAb drug with a theoretic PI of 11. It's highly basic. It's found to have contain variable host cell protein levels in the drug substance samples. For the 5 years batches we manufactured, we found that for 4 of the batches, the HCP level is within 48 to 82 ppm. However, for one of the batch, the HCP level is as high as 123 ppm. It is higher than the arcing limit of 100 ppm. Thus, trigger investigation. So, we decided to not only to characterize the host cell protein in the final drug substance sample, but also look into the host cell protein level in the process in the media to figure out the root cause of the problem. Showing the figure in the bottom left is the purification workflow of mAb. It's a 3-column process. The first column is charge and hydrophobicity-based separation. The second column is charge-based separation, and the third column is hydrophobicity-based separation. Basically, we focus on the host cell protein that identified in the last column eluate and looked into the clearance curve in the formal purification, upstream purification process steps, and then identify the column capability and factor could provide better clearance of this residual host cell protein that detected in the final drug substance sample. So this slide summarizes the host cell protein identification information of 4 samples from the Harvey cell culture fluid to the column 3 eluates. What we found is that a host cell protein called tissue factor pathway inhibitor, or TFPI, it became the most abundant host cell protein in the column 2 and column 3 eluates. And in the column 3 eluate, altogether we can identify 10 host cell proteins. So we plot these 10 identified host cell proteins using their abundance against the theoretic isoelectric point. What we found is that TFPI is with much higher abundance than the other host cell protein, and we also find that TFPI seems to be separated from most of the other host cell proteins based on its electric points. And also, as we know, the protein drug itself has a theoretic PI of 11, so we think maybe there's a chance to separate TFPI out from the drug product through a charge-based separation, basically in the column 2. So here we look into the proteins we identified in the column 2 eluate, and we also plot using the abundance against the isoelectric points. We found that we can detect 30 plus host cell proteins, and these host cell proteins seem to be demonstrating a bipolar distribution. For example, some basic host cell protein centered at PI10 and acidic host cell protein centered at PI6, and TFPI just sitting in between these acidic and acidic host cell proteins. So we take the column 1 eluate and load it on the second column, which is the charge with the column with the mechanism of charge-based separation, and then we use a salt gradient to elute the protein out of the second column and collect 20 fractions. For each fractions, we trace the amount of individual proteins using 1D LC mass. So in this figure, the primary axis is the abundance of individual host cell protein in the unit of microgram per ml, and the second axis is the abundance of the therapeutic protein in the unit of milligram per ml. We found that for the therapeutic protein, it mainly eluted from in between fraction 8 to fraction 14. However, for the host cell proteins, including TFPI, they were either enriched in the acidic fraction or the basic fraction. This kind of pattern, separating pattern, make us think if we can do a hard cut with slight sacrifice on the yield of the therapeutic protein, we can separate out most of the host cell protein, and with that we can significantly reduce the final host cell protein level in our drug substance sample. So at the same time, we also look into the characteristics of TFPI and the potential risk associated with it. So without going into much detail, we believe the presence of TFPI in the final drug substance to be a moderate risk. So now let me switch the gear to the second case study. Is about the Pro A chromatography optimization. As we all know, Pro A chromatography is the most effective step for host cell protein removal for mAbs as well as FC fusion proteins by offering 2 to 3 logs of clearance. Here we compare the host cell protein levels in the Pro A eluate of 8 internal mAb programs. What we found is that the host cell protein in the Pro A eluate were highly variable. For example, from mAb1 is less than 500 ppm, and also for mAb7, the last mAb have an HCP reading as high as 20,000 ppm in the Pro A eluate. And this kind of big difference couldn't be explained by the variation in the titer and the cell viability. Basically, well, we do have a platform per se to manufacture the different type of mAbs. So we decided to design a stacking study to understand this kind of differential host cell protein reduction in the Pro A step So we obtain a null cell supernatant, that is the cell culture fluid without any mAb expression. So we spec in a purified mAb drug substance, and the mixture will allow incubated cell for a short while, then pass through the Pro A chromatography. And we identify the host cell protein in the Pro A eluate by 1D FC mass. So the host cell protein identification information was summarized in the figure on the bottom right. Basically, for one of the mAb, like mAb1, it seems to be very dirty or sticky. The host cell protein level in Pro eluate is as high as 9,000. For mAb4, it's relatively clean or inert. The host cell protein level is below 300 ppm. So we do repeat this wide, big difference in host cell protein reduction when different mAb was studied, although the host cell protein in the null cell supernatant is at the same level. And the result of, we also have the result of blank control, which is not in this shown in this figure, but in the blank control, when no mAb was studied in the null cell supernatant, we were only able to identify 2 host cell proteins. And we think because these host cell proteins have minimum interaction with the Pro A beads, so most of the host cell protein wouldn't survive during Pro A purification. It's more likely that these host cell protein have a strong interaction with the protein drug. So the protein drug serves as a cover protein so that the host cell protein can be detected or survive after the prior purification. So we think the mAb host cell protein interaction is a major reason for different clearance in prior process. So if we look into the process, there are 3 major steps. The first step is we load the mixture of the mAb and host cell protein in your prior column. Then we use a salt wash to try to get rid of as much host cell protein as possible. Then the third step is wash out the mAb using a low pH buffer. So basically, we think this salt wash step could be very critical to disrupt the host cell protein and mAb interaction while maintaining the mAb and Pro A interaction, so that it get the best clearance while not affect the yield of the mAb. So in the figure on the top, so we show one of our studies to try to improve the Pro A white stable by starting 4 different buffers other than the platform process. For buffer 1 and buffer 2 till buffer 4, we found that Overgen performed better than the platform buffer. And also for buffer 1 and buffer 4, if we look at the secondary y-axis, is about the yield of the mAb itself. We found that this kind of input clearance of host cell protein will not affect the yield of the mAb. So that would definitely work much better than our platform process. So in the table below, summarize the top 8 host cell proteins detected when different wash buffers were used. We found that for when platform buffer was used, 8 host cell proteins were detected at significant levels from 84 ppm to up to 2200 ppm. However, when buffer 1 was used, 5 of the host cell protein were below the detection limit, and 3 have significant reduction in the host cell protein level. These 3 host cell proteins named perturbin, actin, and GAPDH seem to be the host cell proteins that are difficult to be removed by the current Pro A process, even though with significant efforts of buffer optimization. So if we look at the shared host cell protein among all the 8 internal programs, we found that these 3 host cell proteins also shared among all the 8 programs. we also found another host cell protein, PLBL2, was shared among 3 programs. PLBL2 is a problematic host cell protein which could be immunogenic and also can have lipid activity and cause strain degradation and impact the drug substance or drug product stability. So for these hard to remove host cell proteins, we think of using an alternative method to reduce the expression levels with cell line engineering. So we decided to knock down instead of knock out this protein because we think each of these proteins may have distinct biological functions. So for each of these proteins, we obtain 3 different siRNA constructs. Then after cell culture of 48 hours, we look into the mRNA level of these proteins. We found that for clustering, each of these siRNA constructs works very well with reduction efficiency of over 80%. For the other host cell protein, we found all of these siRNA worked with one exception of one of siRNA acting not work that well. So we continue for each of these proteins, we select one of the siRNA and keep monitoring the cell culture performance at day 6 and day 10. For clustering, we found that the low mRNA level was retained at day 6 and day 10. So it was for GAP-DH. However, for actin and PLBL2, the mRNA level bounced back at day 10. So basically, this siRNA works for clustering, and GAP-DH, but not for actin and PLBL2. So we want to confirm that kind of this low mRNA level will translate to low protein expression. So we take the day 10, have a cell culture fluid and purify the material by Pro A and look into the host cell protein level in Pro A load and eluate. So we observed for cluster and GAP-DH, there were 80 to 90% of reduction in the Pro A load, and this also translated to a similar level of reduction in the Pro A eluate. So, in summary, as a proof of concept, we found that siRNA knockdown of cluster and GAP-DH were very successful in reducing the level in the Pro A eluate, and in the meantime, the data not shown the cell growth and titer were not affected by into siRNA. So these are the conclusions. Basically, we can. I hope I have proved that by fine-tune the purification process, we can remove some of the lead host cell proteins, and also we can improve the Pro A wash to remove to have advanced host cell protein reduction. For this hard to remove host cell protein, we can also use siRNA technique to knock down their expression level and cause less burden to the downstream purification process. So with that, I would like to thank all people involved in these experiments. Okay, that's it. Now I want to give the control back to Sri.
Thank you very much, Dr. Zang and Dr. Zhu. So just as a reminder to our listeners, if you have any questions for Dr. Lee, Dr. Zhu, or Dr. Tate, then please click on the questions tab and send them to us. We will be asking these at the end of the presentation. I would now like to invite the speaker for the second half of this presentation, Dr. Stephen Tate, to present an industry perspective titled Robust and Sensitive Host Cell Protein Detection with Unbiased SWAT Acquisition. Welcome, Dr. Tate.
Thank you. Thank you for this opportunity. I'd like to thank Biopharma Asia for this opportunity to present some of the work that we're doing in collaboration with the BTI group in Singapore around how to develop robust and sensitive host cell protein methods using our swath acquisition method. So mass spectrometry and host cell protein analysis is increasing its visibility and its importance around the world. And we understand and know as a company that ELISA is generally the method of choice for this, with the understanding that you can get single PPM values for samples and generally on the total proteins that are present within there, and that ELISA is sensitive and relatively easy to perform. Whereas mass spectrometry offers information-rich content, essentially allowing you to provide the quantitation and identification of a vast number of proteins at individual levels instead of in the total. Mass spectrometry is seen as the quantitation method of choice in the small molecule pharma industry, providing high quality data, and has been present there for many years. So we know it has a good basis for quantitation. But one of its big challenges is that it's perceived as a very difficult method and with lack of robustness in the high content assays which are really required for host cell proteins. And this is really driven by the methods which are done within the academic market space, where ultimately it's very low flow and robustness, and so other issues can, let's say, magnify themselves. So the question that we had for ourselves was, can we make mass spectrometry robust enough and reliable enough to generate data of the quality needed to do HCP analysis? So when we started to think about this, we started to think about all the factors that can affect mass spec reproducibility. So it's not just the instrument itself. So it's not just the mass spectrometry or the mass spectrometer and its robustness. It's around the sample preparation. It's around data completeness. It's around the chromatography. So we've been investigating ways of, let's say, improving sample preparation, which we know is time-consuming for mass spectrometry. And so the question was, could we reintroduce robotics into the sample preparation, allowing many more samples to be generated with better robustness than manual procedures? We know mass spectrometers, as I've said before, are perceived as complex. So could we generate equivalent methods to the very low flow methods which are used in the academic, let's say, environments, into the pharma with higher flow rates, which increases their robustness? And we also understand that data processing can be somewhat of an issue. So can we improve areas around here as well? The last area really was data completeness. So this is an issue with mass spectrometers in most of their methods that are used in HCP, is that, as we heard earlier, that they generally will select peptides based on their mass in a single scan, and then do fragmentation for identification. This is a random essentially a random process. And really what we wanted to do was find a way that we could then make this more robust and provide accurate and reproducible quantitation as much as possible on as many, let's say, compounds and proteins that are in there. So we required a non-stochastic method. And then the final question was, could we get this sub 10 ppm or to the single ppm levels for host cell proteins by mass spectrometry? So the typical HCP mass spec workflow involves sample preparation, as I've said, the mass analysis, and then data analysis. The sample preparation is through a protein digestion step, and then MS analysis is generally through DLC separation, followed by the MS analysis of all the eluting peptides. And then data analysis can form two methods really, one around protein identification, and then the final one, which is really what we need for the HCP analysis, which is around the quantitation and how much is present. And underneath these sections here is really the time that we believe at the moment it's currently taking people to do this analysis, or different sets of the analysis. So 1 to 12 hours for the digestion, 1 to 2 hours per sample for the mass spectrometry. And as we're talking quantitation, ideally we need to run replicates here for statistical confidence. And then the data analysis can end up taking days to pull this all together with current systems. So we've been looking at protein digestion and automated digestion and using the Beckman Coulter Biomech robots, and we have systems and kits and methods available which allow us to reproduce, let's say with high confidence, the protein digestion across many samples across many days. And this is being used in both the academic space as well as in many other areas as well. And the idea here is to allow you to digest as many samples as possible with the same, let's say quality that you could get from each other instead of doing it manually, which we know has many different issues. The next step of this really was to improve the chromatography and robust chromatography in complex sample analysis. We know that in the nanoscale, we were able to generate data for many proteins. So we're able to identify many proteins and quantify many proteins. The challenge here is that sensitivity is what we need, but the robustness is not there. We also know that for whole cell protein analysis, samples may not be limited. So we're questioning how can we generate the same quality data at much higher flow rates. And what we've come to the conclusion at the moment is that the microflow LC regime probably offers the better comparison between sensitivity and throughput, and also puts it all into the same industrial setups that are used across many different quantitative applications. When it comes to mass spectrometry, as we heard earlier, there are a number of techniques that can be used. And many of these can be used at different stages of studying host cell proteins. And as you move through, you can come up with the conclusion of what's really the best method as you go forward. So the first method is by far the easiest method, which is down in the bottom left-hand side of the quadrant on the diagram here, which is the MS1. So here you just take the area of the peptide that's eluting and use that as a surrogate for the amount that's present. The challenge is that even though this is really easy to do, it's very difficult to deconvolute those signals in complex samples. And as you start using host cell protein analysis, especially for process development work, there may be many proteins that are present, leading to convolution in the MS1 signal, which is very difficult to remove. The second method as we move around is by far the most sensitive method. So this is the SRM or MRM based method, which is based on triple quadrupoles based technology. It does have a lengthy method development, but that's fine if you're going to use the assay for many, many years, but it also has a very limited coverage. So just like ELISAs, this would be a very focused and very targeted assay where you would not see anything else in the sample appear which you had not built the SMRM method for. The next part around the quadrant here is the MRM-HR. This suffers similarly to the scheduled MRMs in that it has limited coverage, but it has a much easier method development time. These are the most sensitive mass spec methods that you can have, and will offer you the sub-ppm quantitative methods for many proteins and allow you to do internal standard and absolute quantitation methods for them. What we're focusing on is really to use our SWOF-MS methodology. So this is a method which allows us to quantify essentially all proteins which are accessible, or peptides which are accessible to the mass spectrometer. So these are peptides which are within the mass range that we're analyzing, as well as those which are ionized and are detectable by the mass spectrometer. This allows good coverage, but doesn't allow the same sensitivity as the scheduled MRM assay, but allows you to quantify many thousands of peptides and many thousands of proteins. It also has a side part of this which allows a permanent record to be left for the sample. So if you do desire, you can go back and actually reinterrogate the sample over many years to see if new proteins were present or proteins were present that have come up as you move forward through your process development. The SWOF-MS method essentially uses a wide isolation band filter in the first quadrupole. So this is the part of the mass spectrometer which will isolate different compounds, in this case peptides, and in this case we use a wide isolation filter to allow us to cover the whole mass range. But this does mean that there are many different peptides which are co-isolated and fragmented at the same time. What we do know is, as we do this across the mass range, and you can see in this brief animation here, that the actual, let's say, process of the swath, we cover the whole mass range, fragmenting all of the precursors which are within those windows, using either fixed windows, which is displayed here at 25 Dalton widths to cover the whole 400 to 1200 mass range, or with variable windows, which allow us to focus much narrower windows to improve selectivity around specific masses of interest or mass ranges of interest. We're able to then deconvolute these signals in the MSMS that are generated from the peptides. These are used for the quantitation, and we're able to cover this whole mass range and offer MRM type of quantitation within the time that you have for elution of the compounds across the chromatography peak. The typical swath analysis workflow is shown on this slide here. Now, one of the ways of processing swath data is through the use of a spectral library. So we need to have some information about what might actually be in the sample. Pardon me. This doesn't have to be absolute, and it can be over what we believe is in the sample. So in general, what many users who are using swath will do is they will take a stock sample and they will do exhaustive analysis on this to identify as many proteins as possible and peptides as possible within that sample. And this is shown in the top part of this block diagram here. And this uses our IDA analysis, followed by protein identification, searching, and then this step of spectral library creation. This can be single-dimensional chromatography, multi-dimensional chromatography. As you'll see with the work, as I'll describe from BTI, we use many dimensions to get here. Once we have this spectral library, we're then just down to the comprehensive swath analysis. So you'd run your samples with swath analysis. You would use this spectral library of hopefully as many of the proteins that can be expressed from your organism as possible to extract data, and you do this targeted extraction, followed by generating of peptide and protein areas, and then you generate PPM levels. With the work that we're doing with BTI in Singapore, they have been investigating how to generate large libraries for CHO-K1, both intracellular and extracellular proteins. They've used a strategy of using SDS page separation, as well as ultracentrifugation to get different subcellular components, as well as peptide separation using high pH, as well as in the first dimension, as well as running single-dimension samples for both extracellular samples of non-producing and producing cell lines. And all of these have been pushed through the LCMS platform to identify all the peptides and all of the proteins. In doing this, they take the samples and they use standard database surgeon techniques, which are available in many different flavors, to generate a comprehensive list of all the proteins that they can see. And at the moment, the number of proteins which we have in this library, in collaboration with these guys, is around 1,100, and these are highly curated and highly conservative results. And this is from the merged data set of all the different fractions that are present. So we have good coverage of the proteome that's present. And just for example, in a single one-dimensional run, the identification of these from the cellular-based materials around 3,200 proteins. From a protein coverage perspective, because this is important to understand that we need to have good coverage to be able to say that we can accurately quantify these proteins, we have around a median sequence coverage of around 30%, which is very good on all of the proteins which are present. When we take this library and we use it for quantitative measurements, and this is using basically a cellular extract from the non-producing strains that we have available to us, we're able to identify, using the merge library, around 4,200 proteins in that sample, and we're able to get percentage CB measurements on these proteins of less than 10%. In this comparison here, we're showing a single LCMS run to say that the merged library is actually better, so we're able to identify more proteins using this SWOF analysis technique than we can using the single LCMS run. Obviously, the next stage of this is how you determine the PPM values. So we've been investigating different methods from the mass spec data. We understand that the MS or MS1 space using the high-free approach is a standard, well-respected method. But the question was, is there a better way of generating these PPM values using the SWOF data and the MS-MS values? So we prefer to use the MS-MS values because the likelihood of being interfered with is less. And the aggregation of these methods or these measurements to the protein level is somewhat simpler than using the MS values. And also it allows us to have increased statistics around the measurements and the confidence in the results that we have moving forward. We looked at using the aggregate levels, which is the graph on the left-hand side here, against the three most intense, and what we found is that aggregating all of the measurements together to a protein value does give us better reproducibility when we look at standard proteins compared to the three most intense peptides. So with that in mind, we wanted to then study the linearity and the limit of quantitation. So within BTI, they took a series of standard proteins and spiked it into a monoclonal antibody containing solution, and then analyzed this with SWOF analysis. And we aggregated the transition level data up to the protein levels and determined the PPMs. The figures underneath are for 4 of those proteins that were spiked in there, and the red line that you can see on here is the theoretical PPM line, and the blue line is the experimental derived PPM levels. And we can see that there's good correlation between the theoretical lines, and we're also getting down to the 10 sub-10 ppm level, which was very encouraging as a first series of experiments. Since then, we've confirmed the sensitivity level and also increased away from the nanoflow regime, which is what much of this work up to now was shown, to a much higher microflow regime, and we're able to show much better limit of detection down to the sub or even 1 ppm level, as well as good agreement with theoretical ppm levels that we expect for the standards that we're spiking in here. If we look at the linearity of these or some of these proteins, we can see that the linearity, even though there's only 3 points in these graphs, appears to be very good, indicating that we probably have a very good system at the moment for analyzing the level of host cell proteins within the sample. And this is the work that's currently ongoing within BTI to see the utility of this with producing and non-producing strains. So I'd like to stop there and really just say what we're doing with Insights at the moment is trying to see how we can apply the quantitative mass spectrometry to offer a non-biased approach to analyzing the host cell proteins. And coupling that with automation and also robust chromatography to offer sensitivity and a fully robust system which will allow routine low-level ppm analysis. Currently, it looks from the data that we're generating in collaboration with BTI that we're able to analyze the host cell proteins at levels which are required from what we're hearing from other customers in the market segment, which means that we seem to be on the right track. Finally, I'd just like to acknowledge the work that Dr. B and his group in A-STAR, the BTI group there, are doing for us. Without the work they're doing, we would be pretty much in the dark at the moment about the levels that we have, as well as the great work that they're doing at generating the library, which is not an insurmountable or small order of work that they're doing. And of course, I'd like to thank you as well. And I'll hand it back to Sri now.
Thank you very much, Dr. Tate. So at this point, I would like to begin the question and answer session. As I mentioned earlier, if you have any questions for any of our presenters, then please send them through to us via a questions tab at the bottom of your screen, and we'll be happy to answer those. So our first question is, hasn't specifically been directed at anybody, but the question is, at what scale can you do siRNA knockdown, i.e. 10L fermentation, and how effective is the knockdown when done on a large scale? Based on the costs associated with siRNA knockdown, can this be routinely used?
So I just want to make a clarification, right? The use of the silencing RNA in the experiment was done to explore the concept, to see if we could, through locking down the level of the gene expression, to benefit the HCP control of biologics production. So the actual experiment was done on the 125 ml shake flask scale. That's done totally for the proof of concept. Yeah, imagine doing that on the large scale. It will be very costly, and however, through controlling the gene expression was proved to affect the HCP control in the final biologics product. Yeah.
Great. Has anybody else got anything to add to that? No? Okay. So our next question is, can you talk about the chromatographic conditions used for HCP characterization?
Okay. So for all the experiments present in this presentation, we will use our nanofluorescent. Basically, we are using the 25 centimeter 75 micron column with the use of sub 2 micron particle. But recently, we are trying to make a change to our platform LC-MS process. Basically, if you look here, Steve's presentation, basically Sachs promoted the use of microflow by doing microflow, you can have better throughput and higher robustness. And we are also considering to switch back to our conventional ether spray. Internally, we think by using our conventional ether spray, we can further improve the robustness and also throughput. And also because we now have better column choices to enable better column loadings. And in fact, the LOQ we can reach through conventional ether spray is not worse than that from our nanofluorescent.
Great, thank you very much. Our next question is for Dr. Tate. Dr. Tate, is there a specific reason for using IDA for spectral library creation and not SWAP?
Yeah, that's a very good question. And there's a number of people out there who are trying to do direct identification from SWAP data. This is what I would call the cutting edge of SWAP research at the moment, and there are a number of techniques to do that. One that's fairly well respected is the DIA umpire approach from the Nowitzki group. The challenge there is that you still don't seem to get the same level of identification numbers as you do from the IDA or DDA methods. And once you've generated the spectral library, you don't essentially need to go back and regenerate the library for the organism. The spectra and the peptides will always be the same. So at the moment, we're promoting the use of IDA or DDA methods to generate these spectral libraries. And that's also because of the amount of data that's also available in many places for these organisms anyway. But in the future, I can see that, yes, we would do this directly from the SWAP data. It's just not available realistically at this time point.
Thank you very much. Our next question is for Dr. Zang and Dr. Zhu. Could you elaborate on how to tell if an identified protein is a true HCP or a false positive ID?
Okay, and there should be multiple considerations. First, I think if we look on proteomic experiment, it's purely whole cell protein, because the wide dynamic range of the whole cell protein and the protein drug itself is typically more than 5 or 4 or 5 order magnitude. Basically, then if you consider you may have additional immunity or purification step such as 2D LC or depleting, that lower amount of contaminants could be getting enriched and be identified as host cell protein. Basically, we do have good knowledge about common contaminants such as carotene, but how about the other contaminants, basically from the hair, or from the skin, or from the saliva itself, for example. So we better have better understanding of different type of contaminants. And also, Li Zhang mentioned that in case of CHO host cell protein, in case of HEK host cell protein, basically it's very hard to differentiate the protein, the true HCP from the human introduced contaminants. In that case, you would need have a carefully designed control sample. For example, if you do Pro A depletion, you have to have a control sample that could mimic the Pro A depletion process. And another consideration, if you think about the reporting level of host cell protein, the host cell protein amount is nominal to that of drug substance. Basically, because the process itself for the drug substance, basically the yield is usually higher than 90% for each purification step. With that said, the level of host cell protein should also demonstrate a decreasing trend during downstream process. We observed a protein level jump down in a later stage that could likely to be a false positive.
Okay, thank you very much, Dr. Zhu. Our next question is for Dr. Tate. Somebody's asked, would it be possible for you to share your CHOK1 greater than 10,000 protein spectral library?
Yes. So obviously the BTI group are an academic group, that they're not an industrial group, and so they get their continual funding and money from publications. So at the moment we're trying to aggressively, let's say, publish that somewhere so that we can deposit the library essentially into the public domain, and that's the current working plan. But all these things are changeable and flexible, and who knows what the future is. So I do expect to see it available sometime within, let's say, the next period of time, and it all somewhat depends upon getting the publication out from BTI.
Okay, great, thank you very much. So that looks like all the questions that we have today. I would like to take this opportunity now to thank our presenters, Dr. Li Zhang, Dr. Chongfeng Zhu, and Dr. Stephen Tate for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com/webinars. Thank you very much, everyone.
Thank you.